Okay, thank you very much for inviting me. It's a real pleasure to talk here. Can everybody see the slides?
Yeah.
Make sure. Okay, perfect. So I'm going to try and get this under 15 minutes, but do the best we can here. So the first thing I wanted to mention was intrinsic functional connectivity, which we all know quite a lot about. A lot of us actually study this pretty intensely. And one of the main reasons that we study this, of course, is that we think that there are behavioral ramifications of intrinsic functional connectivity. That is that the relative strength of connections between site A and site B are parlayed into something that's behaviorally relevant. And I'm particularly interested in things that are relevant to emotion and affective processing. So this is actually, I'm not sure if people can see my cursor, but this is a paper by Lauren DiNicola and Randy Buckner's lab, just showing that there are subnetworks in the default network that correlate to theory of mind and episodic memory processing. I'm particularly interested in depression, as I mentioned, affective processing and affective neuroscience. And there are, and have been actually for quite some time, network models of depression. So even before intrinsic functional connectivity was the rage, there, Helen Mayberg and others proposed network models such as this. And much more recently, people like Drysdale and the Connor-Liston group came up with these network characterizations of people with major depression and actually sought to differentiate people with depression in terms of their symptomatology and dimensions such as level of anxiety and level of anhedonia, and to correlate that with patterns of intrinsic functional connectivity. So something else that I am interested in, and I work in the TMS lab here with Joan Camperdon, is if these networks have behavioral relevance and they have clinical relevance to depression, how do we directionally modulate them so that we can achieve healthier functional configurations? And there is evidence by Mike Fox and his lab to suggest that where you actually stimulate with respect to the networks matters. Okay, so if you actually stimulate a site that is more anti-correlated to a hub of corticolimbic networks, which is the subgenual cingulate right here, then people tend to do better in terms of their depression. So again, this gives us a sense that when we do TMS in the clinic to treat depression, the site of stimulation with respect to the actual intrinsic functional network architecture of that target matters. So why probe this further? Because we do know that TMS works for depression, but it's not fully effective for depression. If we had a better mechanistic understanding of how TMS changes intrinsic functional network architecture, we might be able to better understand the pathobiology of depression. We might also have a better understanding of normative affective processing and of corticolimbic circuits anchored in the frontal lobe. And just as an aside, there's a lot of practical reasons to do this kind of work because because the dorsolateral prefrontal cortex, which is the site of stimulation for major depression, is one of the most common sites across both investigational studies in TMS and clinical studies in TMS. And it is heteromodal association cortex. So one of the great dilemmas that we have in TMS is that a lot of what we use for stimulation parameters are based on stimulation of primary motor cortex, where there is this readily available neurophysiological biomarker in the form of a motor evoked potential. But what happens when we actually stimulate heteromodal association cortex, which of course is very different cytoarchitecturally from primary motor cortex. So see, these are some of the reasons that motivated this work. And so we could do some studies where we have some form of imaging, which is what we have done in the past, before and after we do stimulation. And in the past, we've actually done protocols where we will have a baseline resting state functional connectivity map. We will do, and the purpose of this, I should mention, is to delineate a functional target for stimulation, and then we'll do a scan before TMS, and then we'll do a scan after TMS. And this was I won't get into this, but this was just a study in which we stimulated the default network with different patterns of TMS stimulation, 1 hertz and 20 hertz. The reason I put this digital clock up here is I like to think of these functional networks as kind of like little red things on these digital clock. By that, I mean that there is a framework that is sort of fixed, that it can't, that these networks can't go out of, right? Just as there are these fixed sort of grooves here. But within that framework, there's multiple functional configurations. So even though we often think of these networks as the default network, the default mode network, the salience network, the frontal parietal control network as being these fixed systems that don't overlap at all, there is a lot of interchange depending on processing demands, and that happens very dynamically. And one of the things that might be cool about TMS, and that we tried to show in this paper, is that TMS might be able to reveal some of these different functional or novel functional configurations that can occur within the topography of these large-scale networks. So the problem is that we can't just do MRI, in my opinion. We can't just look at functional connectivity, because something that's critical is determining what's happening at the site of stimulation. Remember, I mentioned that with stimulation of motor cortex, we can tell pretty well. We have this motor evoked potential, we can tell what's happening. But in heterolimbic association or heteromodal paralimbic association cortex, we really don't have a readout of that. So that made me turn to using the MR-PET scanner, which many of you are familiar with. And so I did the study that I'm talking to you about in Base 6 in the Martinos Center. These are some of the general advantages, and Ciprian Katana has written a lot of this literature about the advantages of MR-PET imaging. I won't go into those, but I really wanted to capture simultaneously the changes at the site of stimulation in terms of the metabolic change, and that's why I turned to the fluorodeoxyglucose tracer, and to correlate that to distributed changes in intrinsic functional connectivity. So this was the design of the study. Very briefly, what we did was a fused MRI PET scan as a baseline for targeting these 2 DLPFC targets. One was a dorsolateral target, which actually is very similar to what we stimulate in the TMS clinic. The other one was a dorsomedial target, which was much more close to the midline. We did a functional MRI scan before, and then we did a MR PET scan after. Why didn't we do an MR PET scan before and after? Well, that's because the half-life of FDG, of 18 FDG, as many of you know, is very long. So that really prevented us from, doing this, the fused MR PET scan before and after. So the FDG comparison, unfortunately, had to be made between this baseline visit and the post-TMS visit on separate days. The injection was done about 5 to 10 minutes before the onset of TMS. So where did we target specifically? Well, because the subgenual cingulate right here, abbreviated as the SGACC here, is a huge hub, and due to some work by Mike Fox that I mentioned, and Helen Mayberg and many, many others delineating this as this critical hub, it has been a target for deep brain stimulation, and so on and so forth. We used functional targeting based on the functional connectivity of the subgenual cingulate. And so what we did was we picked 2 general sites across healthy participants. I did do people with depression, but that's not going to be in this talk because the data is not fully analyzed. But for this talk, we picked these 2 functional targets. One were areas that were most positively correlated with the subgenual, and that's shown by these white circles, and another one that was most negatively correlated with the subgenual, and those are these black circles here. And as you can see, the white circles, these are 3 different network parcellations. So this is the Yo7 network, this is the Yo17, this is the Jonathan Power parcellation. These are all about 9 years old. But as you can see, the positive correlations really fell within the default network. And the negative correlations, we mostly think of those falling in the frontoparietal control network. But as you can see here, this pink and sort of fuchsia color here is the salience network. And so there was a lot of overlap. And one of the things I want to use as kind of a take-home point for everyone is don't ignore this part of the brain. This is a salience network representation that gets a lot of, that gets ignored, I think, a lot. And I'm going to make the argument that it's actually pretty darn important. In addition, some of the other relevant seeds that we looked at, because this medial prefrontal seed is just so huge, also looked at, and I drew this from the Drysdale paper that I showed you earlier, the dorsomedial prefrontal cortex, the ventromedial, the subgenual, of course, mentioned that, and caudate. I'm going to show you something about the caudate. Also, amygdala, which I didn't put in there. So the first thing is just to ask, when we seed the targets, what networks do we get? And sure enough, when we seed the medial, the dorsomedial target here, we get the default network. And so what you see here is these black outlines here are the salience network, the white outlines here are the default network, and it really just beautifully overlaps with those. Interestingly, when we seeded the lateral target, though, we didn't get the frontoparietal control network. We did get a little bit of it here for the network aficionados out there. That is a little bit of that. But a lot of what we got was the salience network. So really, our targets in general, it wasn't perfect. This was more reliable, but in general, the targets are falling within the confines of the salience network. The lateral targets are falling in the salience network. All right. So what are some of the PET imaging results? So interestingly, you know, we all think about, those of us who are in TMS all think about high frequency TMS doing something different than low frequency TMS. Specifically, we think about high frequency TMS as increasing the sort of quote unquote activity of the region that's being stimulated, the region under the coil. And when we look at low frequency TMS, we think of it as inhibiting. For those of you wanting specifics, low frequency would be 1 Hertz TMS or anything below that. High frequency would be 5 Hertz or anything above that. There's also something called theta burst stimulation, which I didn't use in this study, but I theta burst, intermittent theta burst is excitatory, and C or continuous theta burst is thought to be inhibitory. Anyway, so the conventional wisdom is that, well, if you're going to use 20 Hertz TMS, which I did in this study, this is 20 Hertz TMS, big time excitatory, you should be seeing, as you do in motor cortex, increases in activity at the target. And that's absolutely what we found following the lateral stimulation. It was significantly increased versus baseline. But interestingly, for the medial stimulation, the opposite happened. It actually significantly went down compared to baseline. So before we get too excited about this, this is, you know, a relatively small N. It's under 20 subjects. But take it for what it's worth. I don't want to have people think, oh, well, this is now true that different parts of the brain will respond differently to the same pattern of TMS in terms of the metabolism. But I think it warrants us looking at it a little bit more, because we haven't really looked at it too much at all. So this is just showing the comparison between the two. So then what happens with functional connectivity? Well, when you stimulate the dorsomedial target, so this is from the target itself, and again, I included these black and white lines here to show you the default and the salience network. Those are my friends, and those are the ones that reverberate throughout all of my work. So just focus on those two, forget about everything else in the brain. But these are, for what it's worth, these are maps of the entire brain. These are not like specific ROIs. These represent significant changes throughout the entire brain from a specific seed. So from the target, following the dorsomedial stimulation, what you're seeing is, or at least what I'm seeing, is increases with different parts of the salience network, from the target, which is a member of default, to salience. And within the default network, within the sort of stimulated network, so to speak, you see decreases. And this gels actually with a couple of other groups in terms of the directionality of people that have done high frequency, specifically 20 Hertz. It also gels with our prior work where we did a 20 Hertz study that is showing 20 Hertz stimulation decreases functional connectivity within the stimulated network. Who cares? Why does that matter? Well, it matters because if we do studies that are showing that within a given psychiatric disorder, there's increased connectivity and we actually want to disrupt that, we want to functionally uncouple the network, then the directionality matters, right? We want to be able to decrease it. So this is the subgenual cingulate itself. There has actually been a host of articles that have talked about the subgenual cingulate being too functionally tethered to the default network, that there is hyper connectivity between it and the default network. And in fact, following both of these patterns, we actually decreased some connectivity between that and members of the default network. So I want to show you the different seeds throughout the rest of the default network and the salience network. This is PCC, which is one of the main hubs, of course, as you know, in the default network. And again, we're seeing these really dramatic not dramatic decreases, but decreases within the confines of the default network, but really dramatic increases between PCC and salience network nodes. As most of you know, these networks are inherently negatively correlated at baseline. So these networks became less negatively correlated than they were before. So this is following the lateral stimulation. One of the things that I want to emphasize is the lateral stimulation wasn't nearly as powerful in terms of doing this as the medial stimulation was, as you can see here. This is from the retrosplenial node, just showing you some of the same things. This is retrosplenial with a negative. This is from salience network seeds, again, following the medial stimulation. This is the frontal insular seed here, showing, again, increases with default, specifically actually here with a little bit more of these inferior retrosplenial portions. This is following lateral. This is another salient seed. This is a frontal salient seed. And one of the things that I want to emphasize is these frontal salient seeds were some of the only ones in which the lateral stimulation had a punch and kind of compared to the medial stimulation. So this is the lateral stimulation, and looking at, this is actually the homologue of the target on the left and from the right hemisphere seed, showing increases within the opposing network. This is from the caudate, which just an interesting thing, which did really stand out from the lateral stimulation, but not the medial. So this is just a little bit of a summary. Busy slide, but the take-home message is, again, from the functional connectivity standpoint, the biggest thing that we saw was some decreases within the network, and then these crazy increases in connectivity between the default and salience network. Again, just to emphasize, this is these networks being inherently negatively correlated, and then after TMS becoming less negatively correlated. The trend was similar with both the medial and the lateral stimulation. It's just that it was a little more specific with the lateral stimulation, insofar it was kind of isolated to these prefrontal salient seeds. So this is the next part, the next final 5 minutes. I just want to go over this next thing that we did that I'm very excited about. We then asked a question, well, we've looked at FDG at the target, and we've looked at distributed functional connectivity changes. What about the correlation between those? So in this slide, what I'm showing are functional connectivity changes that correlate with changes in FDG at the target. So as I showed you initially, the target for the dorsomedial stimulation decreased its metabolism. So here we're asking what functional connectivity changes went with decreasing metabolism at the target. And in fact, what we see is some of the things that was borne out in the functional connectivity that I talked to you about before. In other words, that when you look at the default network seeds, these are all default network seeds. I include the subgenual cingulate in that because if anything at baseline, it's much more correlated to the default than anything else. It's part of the limbic network. But with all of these default network seeds, what you see are decreases within the default network and increases between the default network and the salience network. These are just some representative seed pairings out here in this outer circle, and the inner circle is just showing a circle map showing the links. So within the default network, these decreases in blue, between default and salience, these increases. So again, these are functional connectivity changes that are topographically specifically changing as the target goes down across participants. Hopefully that's clear, but if it's not, feel more than free to ask questions later. Looking at the lateral stimulation, the same kind of thing actually. So even though we have these divergent patterns of stimulation, or actually I shouldn't say divergent patterns, same pattern, but divergent locations and divergent changes in FDG, we're seeing again this default salience relationship, decrease within, increase between network, that correlated with increasing FDG at the target here at this dorsolateral target. So this is one of my final slides. I think I'm just about out of time, but this is just showing the overlap between the functional connectivity changes that are correlated to changes in FDG at the target, and the functional connectivity changes that were just there in general. In this particular slide, the decreases in functional connectivity are in dark blue here, and the decreases in functional connectivity that correlate with changes in target metabolism are in light blue. And you see here for the dorsomedial stimulation, there is a fair amount of overlap in terms of that. It's not perfect, I should mention. And then these are the increases. When I say it's not perfect, one of the interesting things that I spent a lot of time thinking about, for those who are interested, is there were a couple of C's which really, in terms of the functional connectivity correlation with FDG, really went bonkers. And one of those is the right dorsomedial prefrontal cortex. So this kind of area right around here, as you can see, its functional connectivity with the default network really tanked as the metabolism at the target went down. And interestingly, a couple of people, this isn't just sort of picking out one study that kind of fits my results, but a few studies have actually shown some kind of functional relevance in terms of corticolimbic circuitry in this region. There was a paper by Shalin et al. in PNAS, I think 9 years ago or something like that, or 10 years ago, referring to somewhere this area being like a dorsal nexus. So I thought that was interesting. Another interesting observation was the fact that this trend held true with the subgenual. And then another interesting observation was the sense that the PCC tended to uncouple, PCC being here, with the retrosplenial cortex, particularly in the medial stimulation. And again, these are, so these slides are showing changes between these target seeds and the salience network. And in this case, these are increases. And then I think this might be my final slide. This is just for the lateral stimulation. Difficult to make sense of this, but it was the sort of link between the FDG metabolism and resting state functional connectivity was still very, very strong for the lateral stimulation. But as I showed you before, the functional connectivity, the plain old functional connectivity changes were much weaker and were really relegated to changes between some of the default network seeds and some of the more frontal salient seeds. So it was a little bit more specific as well as the caudate. So I just want to thank the following people for this work, mostly Stephanie McMains, who helped me tremendously with fMRI analysis, David Izquierdo, who helped me with PET analysis. I also want to highlight contributions by the amazing MR-PET staff in Bay 6, and happy to take any questions.